Okay, I think we can start with this seminar, and it's my great pleasure to present my colleague and friend Ramin Massoumi. Ramin is performed his PhD studies at London University in the field of inflammation related to colon cancer. After his PhD studies, he moved to Max Planck Institute in Germany, where he performed his postdoc, and he generated animals. He made a lot of studies on animals. And in 2010, he was funded by ERC Starting Grant. As you know, is a very prestigious grant. And in 2015, he also won another ERC grant, proof of concept grant. At present, he is head of a research group of molecular tumor pathology at Lund University in Sweden. And the overall goal of Massoumi research is to characterize the function of genes in development biology and immunology. He has a large expertise in high throughput drug screening on animal models and using preclinical 3D imaging. I think now he can tell us more about this argument.
I will also use this in case. Okay, hello everybody, and thank you very much, Mario, for this nice introduction, and thank you very much for having me here. It has been a great pleasure to be here and discussing science with some of you. I was thinking, as Mario said, I'm from Lund University, and I was thinking to just showing you where actually Lund is placed. So Lund is placed in the south part of the Sweden, very close to Malmö, and Malmö is actually the third biggest city in Sweden. We are very close to Copenhagen. From Malmö to Copenhagen, there is a bridge, and it takes not more than 20 minutes, so you can go from Malmö to Copenhagen. Up there you have the Gothenburg. This is the second biggest city in Sweden, and a bit further to the north at this side is going to be Stockholm. So from Malmö to Stockholm is approximately 7 hours time. So we are located at a place which is called Medicon Village. This is the largest science park in Scandinavia. So this is the place that the Lund University has been located now. And this is great for us because if you have any ideas and your idea is to developing a drug or screening or anything else, you can jump directly from the university and starting your own business. So the agenda for today's talk is divided in 3 different parts. I'm going to talk about the lung development, and I'm going to show you some of our previous published papers. I will move on and talk about the lung cancer, and there I will show you some of our non-published data, and I will end up with some preclinical in vivo imaging and why it's important to use preclinical imaging. Okay, so let's start with the lungs, which is going to be the focus of this talk. So there are actually some interesting facts that I found about lungs recently, which I didn't know. So first of all, I think all of you know, is the respiratory system is extracting the oxygen from the air and transferring to the blood, and also is releasing the carbon dioxide from the bloodstream. It's a filtration system, and I would say is one of the best filtration systems which is existing. You can think of that as filtrating about 8,000 liters of air, and this removing everything which is on impurities. You might think that the lungs are light, but they are actually weight about 1.5 kilo. So you are going around with 1.5 kilo weight of lungs. And this is something that I didn't know. Actually, the singing helps strengthening your lungs. So basically, I found that when you sing, you engage your muscles. And this is actually promoting the beta breathing and expanding your lung capacity. So if you don't sing in the shower, start doing that, because it's actually going to benefit you for your future. if we look at the development of lungs, it's involved a lot of stretching and expansion of it. And if the lungs could talk and they could brag, they would say that they are the excellent yogis. So they are basically always stretching their to become a fully functional form. Looking at the lung development, it's divided in different steps. And these different steps could be quite complicated. I have been simplified this in 4 different steps. In human, it starts at the week 3, and it's going on until the week 38, and that's the step where you have the fully functional alveoli. So this is what's happening in human. If you compare that to the mouse, exactly a copy of the development is also occurring in mice. The only difference is here, as you would expect it, that this development stage is a bit shorter. So in the mouse, the embryonic starts at the day 9 and continue that until post-development at the age 5. So 5 days after they are born, you will have the fully functional alveoli. And as I said, there are multiple signaling pathways which are involved in regulating the development of lungs, both initiating that, blocking that. And one signaling pathway that we have been having interest to understand whether it has any role in the lung development is a Wnt signaling pathway. So I'm going to tell you briefly about this signaling pathway, which is consisting of a receptor, which in this case is frizzled. You have a transduction cascade of the proteins consisting of Dishweld, the GSK3 beta, axin, APC, and beta catenin. And then you have the transcription factors, which is consisting of TCF and LEF. So when Wnt signaling is not active, so here we have an inactive form of the receptor, what is happening is that the beta catenin becomes phosphorylated by the help of GSK3 beta. This leads to the ubiquitination of beta catenin, and then this leads to the degradation of beta catenin. So this is when the agonist is not present. What is happening is when the Wnt is present, it binds to its receptor, it makes the conformation changes of the receptor, and this basically makes the activation of Dishweld. And the function of Dishweld in this cascade is that it's blocking the GSK3 beta. So GSK3 beta cannot phosphorylate the beta catenin now. So what is happening is that the beta catenin becomes released from the destruction complex, moving into the nuclei, binds to the transcription factor TCF-LEF, and increasing the expression of different target genes. How can you regulate the Wnt signaling pathway? There are different ways to regulate the Wnt signaling pathway, but one pathway that we could identify, this is through function of the protein which is called NLK. So NLK stands for Nemo-like kinase, is a serine threonine kinase, and this is belonging to atypical MAP kinases. So basically, these are the enzymes that need to become phosphorylated in order to become activated. So NLK become autophosphorylated, become activated, and then it can regulate the downstream signaling pathways. And as I said, one of the signaling pathways that we have been discovered recently is it can regulate the Wnt signaling pathway, and this is through by binding to the LEF and make a phosphorylation of LEF. So as I mentioned before, we were interested to know whether the Wnt signaling pathway, and specifically the Nemo-like kinase, has any role in development stages. And trying to understanding that, we generated NLK deficient mice. So these are conventional knockout mice. Basically, you are deleting the NLK in all of the cells, all of the organs. So these are conventional. And we basically observe a striking phenotype. So the phenotype that we could observe was that when these mice were born on the Mendelian ratio, there was no problem with the when they were born. But as soon as 12 hours, we could see that they start to dying. So within 36 hours, not a single knockout mouse was left alone. So all of them were dying then. When we were looking at these animals, after 24 hours or 36 hours, We could actually find out that they looking pale, they are cyanoic, they are shrinking, and they have a smaller body weight. By seeing this phenotype, this made us interested to look at the lung development of the mice because of the cyanoic phenotype that we could observe here. We also looked at other organs of the animals, but we could not see any differences. Looking at the lung, what we found was when we looked at the embryonic day 18.5, so this is basically before the animal are born. When we looked at the alveoli, we found that they have much smaller alveoli in the knockout compared to the wild type. And here you can see a characterization of it. We look at the wall thickness of the alveoli, and we found a significant differences in the knockout compared to the wild type. So from the 18.5, we went for the stages. So we looked at embryonic day 20.5. This is before they are born, and we also looked at the animals 1 day after they were born, and we were isolating the lung, and we were looking at the lung tissues. And what we could observe was that from 18.5 until P1, we have a dense amount of the cells in the alveoli. So basically, we don't really see the shape of the alveoli that usually you see in the normal cases. We looked at different parameters. We looked at the cell survival. We looked at the apoptotic cells, necrotic cells, but we could not really see any differences. So the only differences that we could observe was based on the proliferation. So here we use 3 different proliferation markers. We have been using the BRDU, we have been using Ki67, and we have been using the cyclin D1. And basically in all of the 3 cases, we can observe a significant differences in the proliferation rate of the cells in the knockout compared to the wild type. So these are the epithelial cells which are proliferating extremely fast. So just to summarizing this part, I will not go into details. If you are interested in this study, you can read it. This was published many years back, 2016. But the outcome that we found was that if you make the deletion of NLK conventional deletion, you will have the thick, smaller alveoli, and they have an irregular morphology and hyperplasia. Since we have the phenotype of the knockout in the lungs, we were then interested to find out what does it happen if you specifically, in the organ specifically, deleting the NLK. And here we have been publishing a few articles where we're targeting specifically in the tissue-specific sites. For example, in the fibroblast, we have been deleting the NLK, or in the T cells. So in the T cells, what we could find was that if you delete the NLK from the T cells, we have a reduced number of CD8 cells. I also forgot to mention that we have ongoing work that we are deleting the NLK in the lung, and we are looking at the adenocarcinoma, and it looks like, as we were expecting, NLK acting as a tumor suppressor gene, even in adenocarcinoma. But this is ongoing work, and we are hoping that we can publish this study quite soon. Okay, so let's move to the lung cancer, and what I'm going to share with you is some of our unpublished data. I start with some facts about the lung cancer. If you look at all of the incidences for all of the cancers, which is about 19.3 million new cases, the lung cancer is among the top. So lung and the female breast cancer are 2 of those which are among the top. Looking at all of the mortality of the all type of the cancer, the lung cancer is in the top with the 18%. So 18% of 10 million deaths is caused by the lung cancer. Looking at the types of the lung cancer, so lung cancer can be divided into the non-small lung cancer or small cell lung cancer. And further on, the non-small lung cancer can be divided in 3 different, so it can be either adenocarcinoma, squamous cell carcinoma, or large cell carcinoma. And the one that is quite highly affected patients is actually adenocarcinoma. So adenocarcinoma is among the top lung cancer which is existing. Looking at some molecular classification of adenocarcinoma, what has been shown that both at the early stage, but also at the metastatic stage of the adenocarcinoma, the KRAS mutation is one of the top of the genes that is mutated. So we have 30% in the early stage, while in the metastatic, this number is also about 30%. So the KRAS is one of the drivers of adenocarcinoma. Beside the KRAS, the P53 is also another driver mutations. So KRAS and P53, either alone or in a combination, are the driver for adenocarcinoma. Very recently, it has been coming out many papers. I have been taking 2 examples that actually Wnt signaling pathway is also one of the drivers. And what has been shown, which is interesting in these 2 studies, that actually Wnt signaling pathway is promoting the KRAS and P53 mutation, that the tumors is becoming more aggressive. They can metastasize much faster. And also looking at the top driver genes, you can also find the beta-catenin. So this is the stabilized form of beta-catenin, which is among the driver genes. So here we talk about the P53, the KRAS, and the beta-catenin that we have been focusing on. One part, which is what has been occurred during the lung cancer therapy, is that from beginning, all of the lung cancer patients was harboring the modern conventional therapy. So basically, all of the lung cancer would receiving the same therapy. What was occurring then was that in all of these patients that were treated, you would have some responder, those which are responding well to the therapy. You will have some patients which are not responding, and we would have a patients which have the side effect. Very recently, this has been changed now. So now, the treatment of the non-cell lung cancer is totally different from small cell lung cancer. And even more, the treatment for these 3 types are quite different. But we are not there yet. What we would like to have a precision medicine. Precision medicine means that you would like to have the selected treatment for the selected patients. But the challenge here is to find a strategy to identify which drug candidate you should be able to use that for this targeted therapy. So what we have been focusing on, 2 different strategies. So these 2 strategies are either 3D organoids and also the preclinical imaging. And of course, would guess that you can use many other protocols, many other strategies, but we were interested to applying these 2 strategies. And what we are hoping that these 2 strategies can actually make it quite fast, that you can move from the preclinical step to the clinical studies. Okay, so let me just give you some background about the organoids. So the application of organoids has been blooming a lot. There are many labs who are working with organoids. They can be used in different applications. Here are some examples. It can be used for drug discovery, toxicology. I already mentioned precision medicine, disease modeling, development, and etc. So what we were interested then, to take the knowledge from the adenocarcinoma, where you have the KRAS mutation, where you have the P53 mutation, where you have the Wnt signaling pathway, and a combination of those, and trying to understand how the organoids can be applied for the in vivo models. So these are the combination of the animals that we have been using for inducing lung adenocarcinoma. And as a control, we have been using the healthy mouse. So the healthy means basically that we have been isolating the lung tissue from these mice. So for the results that I'm showing to you is going to be either KRAS alone, KRAS in combination with P53, or KRAS in combination with both P53 and beta-catenin. And that's what combination means here. The way that we have been doing that this mice is developing lung adenocarcinoma. We are making the organoids, we are characterizing these organoids, and we are making the drug screening. Just to mention that for development of lung adenocarcinoma, for the KRAS, it takes approximately 8 to 9 months, Combination KRAS to p53, it takes approximately 6 months before we can see the first adenocarcinoma. And when we have a combination of the KRAS, p53, and beta-catenin, this goes quite fast. So within a 4 to 5 months, we have the first adenocarcinomas. Okay, so we generated adenocarcinoma. We isolated the tumors or the healthy tissues from these tumors. And as you see, they have a typical organoid structure, roundish types. We do not really see any differences if you have a KRAS alone or if you have a KRAS comparison to KRAS with the p53 or KRAS with the beta-catenin. The only differences that we see, just looking at the structure of cells, is that suddenly when we have beta-catenin included there, the cells are not roundish anymore. So now they are misshaped. And to be honest, we don't really know that why. We have been investigating. We have been looking at the different cytoskeleton parameters. We have been looking at the actin filaments. We have been looking at the microtubules. But we don't really see any differences there. So this is something that we are still searching for. But one could speculate that, as I said before, the Wnt signaling pathway together with the KRAS p53 can make the cells to become more metastatic, that this is that the cells have now the prone to become metastatic cells. We have been generating the organoids. We are making the biobank. This number has been increasing now to 40. I'll show you here 20 only. What should I also mention that there are some of the adenocarcinoma which is metastasizing, and we are also isolating these metastatic tumors. So here, for example, we have organoids from the adenocarcinoma which has been metastasized to the liver. So we are isolating that, and we are making the same organoids. We have been performing the histological characterization of the organoids, and these are the simple way that you do the HA staining. And for us, it's important to comparing the tissue with the organoids so that they have the similar shape, the similar characterization. And you see the healthy tissues, the p53 mutation, the KRAS with the p53, and this is the liver metastasis of the organoids. We have been also looking at different parameters. We have been looking at the thyroid transcription factor 1, which is only expressed in adenocarcinoma, because this is very important for us to be able to show that our organoids is actually a clear adenocarcinoma and not healthy tissue is actually occurring there. So you see in the tissues from the mouse isolated, you see very nicely the expression of TTF1, and you see the same expression in our organoids. Another marker specific for adenocarcinoma is CK7. Again, we see that in the tissue isolated from the mice. We can also show the same tissue in the organoids. Another interesting part is that we have been looking at PAS staining. Again, the KRAS p53, and KRAS p53 and beta-catenin. And what is interesting in the past that we see clear differences when we have the triple mutant. In the triple mutant, we have a high level of PAS. So the expression of the PAS is increased. This could have the suggestion that these are actually mostly affected from the mucin. So this could be the mucin-generated cells. What we also have been performing is that the organoids, which we have them in the culture, they have been transplanted back into the mice. And here on the left side, we have been transplanting the KRAS p53 cells. On the right side, we have been transplanting the healthy cells. And as you see here, you see a tumor which is growing extremely fast in the double mutant, and we do not see any growth of the healthy cells, which means that these cells can be regenerated. In the same way, we have been looking at the left side, the KRAS, p53, and beta-catenin, and on the right side, we have been taking one of these liver metastasis tumor and transplanting that. And what I have to mention that these cells are actually invading extremely fast in this animal. They are metastasizing extremely fast. Okay, so after the characterization, what we wanted to know is trying to find out drugs that could actually be involved for the KRAS, P50, and beta-catenin and block the activation of the organoids or adenocarcinoma which harbor these mutations. And we decided to focus on two different parameters. We decided to focus on the kinase signaling. And the kinase signaling, so the screening that we have done, so the library is containing inhibitors against the tyrosine kinases, MAP kinase, and PI3 kinase. And we also made a high throughput screening on the epigenetic changes. And in this case, we have been using the drugs, small epigenetic, for the histone modifications and the DNA methylation. And why combining now epigenetic changes with the KRAS? There has been many papers which has been published very recently about the effect of the KRAS for epigenetic changes. I have been taking 3 examples here. So in the first of the study, it has been shown that the oncogenic KRAS expression induces the DNA methylation. And the opposite has also been observed, that epigenetic dysregulation now induces the KRAS activation. And finally, in the pancreatic cancer, it has been shown that the oncogenic KRAS expression actually caused the acetylation. So basically, there is a clear connection between the KRAS and epigenetic changes. So I'm going to show you just one example of a set of the high throughput screening that we have been performing. So here you can see the pathways. You can see what are the drugs that we have been using, what do they targeting, and what we were interested in to see the maximum inhibition. So +3, for example, +2, +3 in the double mutant, and we would like to have no inhibition in the healthy lung because we don't want to have any adverse effect. We don't want to have any toxicity there. So by doing that, by doing the high throughput, we selected 3 drugs that we did further investigation. And I'm going to show you just one example about doing the IC50 of these 3 drugs. And in all of the drugs, we have done the high throughput screening for the KRAS, P53, KRAS, P53, and beta-catenin, and also for the liver metastasis. And what we are observing by using these 3 drugs is that suddenly the IC50 is become much lower when we are using the triple mutant. So it seems that they are quite sensitive. We went on and we also did a high throughput screening for epigenetic changes. And here we selected 3 of those. I think you are all familiar about the 5-ASA. So this is a demethylation agent which is used in the clinic. The 5F as well, but we decided to go on with a drug which called decitabine. So this drug is actually causing inhibition of DNA methyltransferase. And as I showed you previously with the other drug, with the decitabine, we also see the similar effect when we are looking at the IC50. So the IC50 for the KRAS P53 is much higher when we compare to the triple mutant. And of course, the obvious question was that do we see any synergetic effect when we are combining these 2 drugs? And the answer is yes. So here we have been combining the PKC 412 with the decitabine, and we see a synergetic effect which is about 5. So we were quite happy about these results. We are also receiving samples from the hospital which we are collaborating with, and the samples that we are receiving from the hospital, they are either the fine needle aspiration, and usually we don't really get so much materials since they are aspiration, but we are also receiving the biopsies, and we are doing exactly the same way. These materials has been dissociated. They are used in the 3 cultures, and we are making the organoids. I'm going to show you 2 examples of the human samples materials that we could make the organoids of these tissues. There are also other examples, and beside adenocarcinoma, we are also receiving squamous cell carcinoma, and again, in the same procedure, we could make an organoids of those. And the obvious question would be, again, to be sure that these are generating from the adenocarcinoma and they differ from squamous cell carcinoma. So And one example is TTF1. When we are applying that on a squamous cell carcinoma, we don't really see any expression of the TTF1, which says that it's coming from the squamous cell carcinoma, while when we are looking at adenocarcinoma, you see the expression of TTF1. And the whole idea with this patient's material sampling is that we are selecting out the patients which has a mutation in the KRAS, in the P53, and if you are lucky also with the beta-catenin, that we can use this for further analysis. Okay, so let's move on to the last part of this talk, which is about the preclinical in vivo imaging. Just very shortly about imaging, why it is important. There are many different benefits of imaging. Here I have been highlighting a few of those. It's non-invasive. You don't need to do any surgery when you are doing the imaging. You can visualize in the disease. You can monitor in the treatment of the response. You can find the health condition. You can use it for prognostic predictive info. And this is the rapid investigation. So there are different preclinical imaging techniques which are existing, but those which are existing for animals, these are shown here. So you can either have PET together with a CT scan, you can have a SPECT with a CT, you can have an MRI, IVs, micro-CT, or photoacoustic. So these are those that can be used for the small animals, so either mice or rats. So for the rest of my talk, I'm going to focus on IVs, and I'm going to tell you why it's important to use the IVs for the drug screening and also as a preclinical imaging. So a few facts about IVs. So by using IVs, you can have a real-time monitoring of your subject. You can make the full body screening of it. You can image up to 5 animals at the same time, so it's saving a lot of time. And what is important is that you can visualize in the living cells and tissues. So in order to use IVs, you need to have some optical reporters or probes. This could be, for example, the luciferase, it could be fluorescent proteins, it could be nanoparticles, it could be quantum dots, it could be fluorescent dyes, label proteins, label cells, label bacteria, or anything else. So you need to have something which is labeled when you transplant into the mice or rats. Other benefits of the IVs is the following. So this basically shows a simple type of the experiment that you have a mouse, you make the disease into the animal, you treat the animals, and you're collecting the data. The problem with this traditional work of this type of the study is that you're totally blinded. You do not really observe the disease development in the mice, and you do not really know what does it happen for your treatment. Is your treatment effective or not? The only thing you are relying on is that you have to wait until the ending step. This is the ending step that you have to endpoint, collect the data, and find out whether now your treatment gave any effect or not. So by using IVs, you can collect the data at each step already from beginning. You can follow the disease progression, and you will also know when is the critical time for using your therapy. I'm going to show you an example about using IVs corresponding for not using IVs. So here there are a few cancer cells which has been transplanted under the skin of the mice. Okay, so if you would measure the growth of the tumors by using the caliper or ladder, basically until 24 days, you would not be able to measure anything because nothing is visualized there. You would be able to see something after 28 days. You would be able to measure that something's growing, and suddenly after 35 or 36 days, it's bang, it's just blooming up, it's just growing fast. However, if you would use IVs, which in this case with only 5 cells, you can visualize that already after 7 days. So you can get the value after the 7 days. And in the same animal, so this is very important that this is the same animal that you are looking at it. Same animal, you can look at the day 7, day 14, and you can monitoring how this tumor is growing. And you can get a graph which is totally different from when you are not visualizing that. Other part which is very important is that when you are not seeing, It could be that you are measuring the dead cells. It could be that you are measuring the edema, and it could be that you are measuring the necrotic cells. In this scenario, you are only measuring the living cells. Other benefits is the selection process. So in the usual cases is that you have a bunch of the animals, you have a group of the animals, let's say you have 10 of the animals that you're starting with. And the benefit of the IVIS is that when you're inducing the disease, you can actually selecting out those animals which harbor the disease. So the rest of the animals will not go through your treatment. You're only selecting out, and you're only looking at those animals which having your disease. You are treating those animals, and you're looking at the real-time monitoring. So I'm going to show you a few other examples. So this is the same animal again, transplanted the tumor cells in the 2D. You can see how the tumor is growing. And after a few weeks, you can see that the tumor has been metastasizing now. You can get a value of how the tumor is growing in this scenario. You can make a 3D imaging, and by doing the 3D imaging, you can find out where the tumor has been metastasizing, which organ has been metastasizing. In this scenario, the tumor has been metastasizing to the brain, that you can see it. You can do the ex vivo 2D imaging, so you can basically open up the cavity of the animals, you can detect in your cancer cells, and if you're interested, you can isolate in your cancer cells, put them in petri dishes, and you can use them for the further analysis. Another example that I'm going to show to you, so these are the pancreatic cancer cells which we have been transplanting into the pancreas of the mice. I just have to say that this is a complicated surgery because the pancreas of the mouse is not so large. So what we have done is that we have been injecting human pancreatic cells into the pancreas, and for us was very important to be sure that the surgery was success, and for us was very important to know that the pancreatic cells are actually in the pancreas. And by using the 3D imaging, we could be sure that the cancer cells, they are actually located into the right site. And I have to just emphasize that this is only 6 hours. So after the surgery, we make the mouse to sleep, and we can be sure that the cells are in the right place. And we could monitor in this mouse for many, many weeks. I show you the results after 8 weeks. Suddenly from the pancreas, this tumor has been metastasized into the different organs, and we can basically localize the different organs. So again, you can collect the data at each step. You can follow the disease progression. You can monitor in the same animal, and this basically make that you don't need to a bunch of the animals. So this reduce and refine. Another example is that we have been using the breast cancer cells, injecting into the breast of the mice, and after a few weeks, suddenly these cancer cells has been metastasized into different organs, which we can use a CT scan, and then further on, we can find out exactly which organ in the animal that this metastasizing has been located to. Okay, but what if now? So what I have been what I have been mentioning until now is that you can follow the tumor growth. But what if you can add extra information on the top of it? So the extra information means that you can looking at some biological aspects of it. So what we have done in this scenario is that, again, we transplanted the tumor cells, and we can localize the tumor cells, and we can look at the growth of the tumor cells. But at the same time, we have been using the fluorescent proteins, and we have been looking at the vascularization. So this is the same animal. While you are looking how the tumor is growing, you can also look at the vascularization at the same time. So you can extract much more information regarding that. So why is it important to understand these biological changes? This is how the disease initiation goes to the early disease symptoms. You have a subclinical disease, clinical disease, and you will end up with the endpoints. And basically, if your therapy has been success, this will lead to disease inhibition. So basically, it means that you have been effective treatment. However, if your treatment is not success, this will lead to the chronic disease, so ineffective treatment. And again, I guess most of us is focusing on endpoint. Is the endpoint that we are taking out the tissues? Is the endpoint that we are looking at the apoptotic cells? Is the endpoint we are looking at the Ki67, at the proliferation cells? What if now you can actually add extra information already from beginning of the stages, already at the disease initiation, you can add some information. And what are the information that you can extract? There are many different information that you can extract. You can extract the metabolism. You can look at the angiogenesis. You can look at the early toxicity. You don't need to wait until the end point and looking at the toxicity of your drug. You can already in the beginning, in the disease initiation, you can get an idea whether your drug is giving the early tox. And also inflammation and vascular leakage. Okay, I will go quickly through this. So the application area by using the IVs, the oncology, you can use the PDX cells, CDX, you can track the cells as I show, you can follow the route of the metastasis, different application on the diagnostics and different treatment that can be applied. So just to summarizing about the imaging for the drug development, we feel that you get a 360-degree scope of the treatment. We feel that, as I said, this is the most cost efficient. You don't really need to have a bunch of the animals that you can use there. You can monitoring many mice at the same time, 5 mice at the same time. This is a time saving. And as I said before, you don't really need to have a bunch of the subjects to analyze, and this basically lead to the reduced number of subjects. So just to summarizing, we need to study and discover effective drugs as well as more patient-focused models in preclinical studies. And what we are suggesting is that 3D organoids, drug screening with the preclinical imaging together can facilitating this road, at least some part of it. By that, I would like to end, and I will acknowledge all of the people in my lab who has been actually doing the work, our collaborators, and our foundations. And thank you very much for your time.
Thank you, Ramin, for this very clear presentation, a very interesting presentation. There is some question also from Zoom audience. Nando?
Thanks for the talk. Very impressive, especially for the imaging techniques you showed. It's just mine is just a curiosity. Is there a way to integrate imaging data with, for example, single-cell transcriptomics?
Yeah, it's an excellent question. As I show you to you 5 cells, labeling cell 5 cells. I just have to say that these cells were unique cells. They were highly expressing the luciferase. Usually, in order to see a signal, we need to be in the range of 100 to 200 cells. So I think the technology is not there yet to be able to make a single cell yet. What can be done, as I said, you can make the ex vivo, after ex vivo extracting out, and from there you can do the single cell expression. But excellent question, yes.
I have here questions about the 3 mutations that they use to create organoids. I was wondering if this combination of mutations is frequent in patients with lung cancer.
Yeah, I mean, is about, so if you talk about adenocarcinoma, yeah, so there we have about 40%, which was the KRAS mutation. And then these numbers become a bit lower when you have beta P53, and then when we have beta catenin, it's become a bit reduced. But what we are doing now is that we are collaborating with our facility just to extracting out this data, because there is not so much data about this combination of those 3. So this is something that we have to wait and see. Yes.
It would be very nice to see how the phenotype of patients with these 3 mutations.
Exactly. So that's what I'm hoping, that at least we have some percentage of the patients which are harboring that, but still for our road, I would say that what we are finding is that it could be for the KRAS alone, it could be for the KRAS P53, and KRAS P3 and beta catenin.
Is there any other question? Okay. Thank you again for this very great talk, and I think we can conclude this seminar, and thank you for your attention.